All right, I am going to discuss Chapter 13, Section 3, Mendel's Law of Segregation. In your textbook, this actually starts going into Punnett squares, and I've discussed that in um, previous videos. I'll hit on it briefly just as a quick review for those of you that might need it, but I think generally most people find Punnett squares rather easy. So this starts talking about meiosis, and we talked about meiosis in chapter 12. And if you remember, meiosis is how sperm and egg cells are made, how the gametes are made. Um, Remember that the cells produced during meiosis are haploid. So the alleles for a particular trait, if a person is homozygous dominant, um, as is mentioned here, so homozygous dominant, all their gametes, they have two copies of the dominant allele. So all of their gametes are going to have the dominant allele for that gene. And we see the same thing with homozygous recessive. So if they are homozygous recessive, they have two copies of the recessive. So the only gamete they can have are the recessive allele. However, if a particular if we have it's talking about the flowers again. If we have a purple flower that is heterozygous, then it's going to separate. So they could either give a dominant or a recessive allele. And let's kind of look at that a little bit more detail here with the picture. I like this picture a little bit here. It's showing you this and P is not the best letter for this in this case. So this is our homozygous dominant. I'm going to put A's over here just so that you remember that this one is the homozygous dominant and this one is the homozygous recessive. They used P, whatever. It just, to me, is easier. Again, I will never stop saying this whenever I teach genetics, that it's just easier to use letters that look very distinct, capital and lowercase, so you can see dominant versus recessive. So this walks you through the DNA replication. So this is this is our starting cell, our germ cell for both of these. And there's DNA replication, so now we double the number in that. So now instead of having 2 chromatids, we have 4 chromatids in each cell. Then through one division, we have half go into this cell, half go into this cell. Same thing over here with our homozygous recessive. It's the same thing happening on both sides. It's just we have 2 different alleles. And then at the end of our second division, remember meiosis divides twice, we have a single chromatid that carries, in this case, the dominant allele. And again, this homozygous dominant individual can only give a homozygous or a dominant allele because they have 2 copies of it. That's all they can give. So all of their gametes are going to have that dominant allele. And when we look at the homozygous recessive individual, they have 2 copies of the recessive allele. So all of their gametes are going to end up having the recessive allele for this particular trait. Let's say it was the flower color because that's what we keep talking about with Mendel's pea plants. If these 2 individuals have offspring, if we were to breed, let's say this purple and this white flower, so this would be our purple flower here, and this would be white. Well, this one's giving a dominant allele, this one's giving a recessive allele, so we're going to actually have offspring that are heterozygous for this trait and are going to have purple flowers. Let me clear this off the screen so I can scroll up. This is just, kind of showing this. And again, it goes over how to do Punnett squares. If you kind of got lost, kind of got stuck when I did this before, this is kind of a good place to look. There's some good vocabulary here. It talks about a test cross. If we have an individual who has a dominant trait, let's say purple flowers, and we kind of did this if you looked at the examples previously. or in the PowerPoint that I did. I'm silencing my phone because I forgot to do that before I started this video, and I don't want to restart. So we can do a test cross, and we actually kind of did this in practice problem number 3, or practice question number 3, in my video going over the Mendelian genetics PowerPoint. We knew that there was purple flowers, and we can figure out based on a test cross with looking at the offspring. If you cross somebody with a dominant trait with somebody who has the recessive trait, because the recessive has to have 2 copies of that allele. We know that. Because again, in order for the recessive phenotype to show up, you have to have 2 copies of that allele. So it makes it easier to figure out what the genotype of the dominant trait is, or what the parent is for that dominant trait. So we can look at that. I already talked about monohybrid crosses before, mono meaning 1. So we talk about monohybrid crosses. Kind of goes into this. There was a whole bunch, and I mentioned this before. So there were 7 traits that Mendel looked at for this. So in order to determine this, he had to do a bunch of test crosses. He had to take round and wrinkled and breed them together and see which ones came out when. And this wouldn't be done with one generation. This would be done over multiple generations. Same thing with yellow versus green. This is how he determined, the pod texture, smooth versus wrinkled, pod color, green versus yellow, flower color, purple, white, where the flowers are, if they're short or they're tall, stem length. So these were all different traits that he looked at. But you got to remember that when he was doing these tests, he was figuring this out as he went along based on his observations. So in order to figure out that the flower position along the stem was dominant at the tip, he would have crossed these together to see what happens, to see what the results were, and crossed them again and made more and more observations based on this, based on his observations and recorded data, and repeating the results over and over again. I got a little distracted there. So really what we're talking about with the law of segregation, I'm going to go back to the point of this whole section, is that these alleles separate. So it goes back to meiosis. That's the whole purpose of me talking about meiosis. Got a little wordy. I think they kind of got a little crazy with their vocab here, and I was just kind of going with what they had. But really, the law of segregation in genetics talks about, you have a pair of alleles, and they're going to be separated. So yes, we used example of a homozygous dominant and homozygous recessive parent, but if we looked at even the heterozygous parent, if we have the heterozygous, I'm just going to use the P's that are right here. Again, I don't like that, but whatever, it's there. So you have one capital, one lowercase, one dominant, one recessive. These are going to separate. If we were to take this parent or this, this plant and cross it with another plant, this parent is either going to give a dominant allele or a recessive allele to their offspring. And that is where the law of segregation comes into play. They separate. These alleles are not inherited together. And that goes back to when we talked about meiosis being haploid, or producing haploid cells. This is the full set. This would be diploid, having 2 copies of the allele. But haploid would only be having 1 copy. So that when a half and a half come together, we have a whole. And again, we talked about that when we did meiosis. And that's just where this is, kind of taking the previous information that we learned and applying it, and how it's relevant and how it's important. We can look at this figure out here. It says, so how many genotypes are possible in the F2 generation if P is dominant, R is recessive, purple is dominant, white is recessive. And it's actually kind of showing you how they're going. If you have a homozygous purple and a homozygous white plant, well, again, the only thing the purple parent can give, they have two copies of the dominant allele, so they're giving all their offspring a dominant allele. Same thing for the homozygous white plant. They have two copies of the recessive allele, so all of their offspring are going to get a copy of the recessive allele. So the resulting offspring are actually gonna, they're gonna be called a hybrid, where they're gonna have, it's heterozygous. So they're gonna have one dominant and one recessive allele, because the only thing one parent can give is a dominant allele. The only thing the other parent can give is a recessive allele. So they're all going to be heterozygous. And we can actually do, a Punnett square, and we could show that. But actually what it shows on this one is if we take those resulting offspring, those from here. So this is we call this the F1 generation. So this first one is actually referred to as like the P1. And then this one is called the F1 generation. So the first generation of those parents. And then if we cross those and make a third generation, we'd have the F2 generation. And you can see the Punnett square here. If we take two offspring from this generation and cross them, so we have two heterozygote individuals, big P, little p, crossed with a big P, little p, then we can see that white flower pops up. And this is what Mendel actually noticed. He took, a purple and a white, and he mixed, sperm from, pollen from one to the other, and had all his resulting offspring were then purple. And then he took two from that and was able to see that the white flower came back in that next generation. It seemingly disappeared in the F1, but it reappeared in the F2 generation. And this is just kind of talking about Punnett squares and how that works out. I mentioned this before. It's getting a little bit more into the Punnett squares. It's a great read if you need some help with it. So basically, again, I'm going to restate the law of segregation says that the two alleles that the parents have are going to separate. So if it's the flower color, they're going to separate. If the parent has two dominant alleles for that, well, the only thing they can give is a dominant allele. They're still going to separate, but the only thing they can give is dominant. The best example is if they're heterozygous for the trait. They're not passing on a dominant and a recessive to all their offspring. They're passing on a dominant or a recessive allele to their offspring. I hope this helped a little bit understanding section 13.3.